Welcome to the second talk series on genetics and evolution, organized by the Journal of Genetics. We are very pleased to deliver this talk. Professor Pannell is a professor of plant evolution at the Department of Ecology and Evolution at the University of Lausanne, Switzerland. He carried out his doctoral work at the University of Oxford on the evolution of combined sexes in plants. Following postdoctoral research with Spencer Barrett at the University of Toronto and with Brian and Deborah Charlesworth at the University of Oxford as a lecturer in 1999 and went on to become reader and professor. University of Lausanne from 2011. Professor Pannell has been interested in the evolution of dimorphism, sex allocation strategies, various aspects of polyploidy, and in the of metapopulations, and has published important papers in all these areas. Today, he will talk to us about sex ratios, sex allocation, and transitions between hermaphroditism and dioecy.
Thank you very much, Vidya, for your kind introduction, and thank you to all the organizers for the invitation to present some of the work we're doing here in Lausanne. What I'd like to do in this seminar is to raise a few questions about how plants use their limited resources through for their reproduction through male and female functions, and perhaps in so doing, introduce to many people who are not used to thinking about plant evolution and the plant reproductive strategy, some of the interesting questions that that come up when looking at how plants flower and the different floral strategies. As most people will be aware, the vast majority of plants are hermaphrodites, and that has been quite a useful point for demographers over the years, because each and every plant in a population produces seeds, and so the idea that one can consider demographic properties in terms of just the reproduction of each plant has been very useful. However, I think this has led to what one might call a seed-centric view. In recent years, this perspective has been overturned, but for a long time, people focused very much on considering the plant from the point of view of its female function. In other words, the production of seeds, and the male function was largely forgotten. And I think this is perhaps this quote from John Harper, who was very influential in the early part of my career, in a book he wrote on plant population ecology. He noted that a plant is the only means with which a seed produces more seeds. And I think this encapsulates this seed-centric view, whereby plants are really basically there to produce seeds. And actually, it's quite interesting to think that this book was published just a year after Richard Dawkins's book on the selfish gene, which of course considered organisms as the only means by which genes propagate themselves. But of course, hermaphroditic plants don't only produce seeds when they make their flowers, and we need to consider the importance of the male function. What are the implications of the male function in plants for the evolution of reproductive strategies in general? Because in the end, every individual has a mother and a father, which means that the fitness benefits through male and female functions are exactly equal in any population. And so by only focusing on the female function, one is potentially missing half the story, and perhaps a very important part of that story. And so for the rest of my seminar, I'm going to be focusing on this question as to how plants should allocate their resources between their male and their female functions, what the implications of the possibility of plasticity in sex allocation are, where a hermaphroditic plant, for example, can adjust its allocation to its male function versus its female function. And specifically also towards the end of the talk, considering transitions between combined sexes, where plants have both male and female function, the hermaphroditic case, and separate sexes, what we call dioecy, where plants specialize as being only male or only female. And that transition has happened a great number of times in angiosperm evolution, the evolution of flowering plants. Thousands of times we see transitions from hermaphroditism to separate sexes, to dioecy. And that has immediate implications in a number of areas, which I'll touch on later in my talk. One of them, obviously, is the evolution of a sex determination mechanism. How do plants decide to be male versus female? Another is the opportunity then, once plants have evolved separate sexes, to express differences in their morphology and behavior that go beyond just the production of the male and female flowers. In other words, the evolution of secondary sexual dimorphisms, which are very widespread in plants with separate sexes. If we think about the evolution of sex allocation as a type of sex ratio question, this gives us a nice inroad into some of the questions which will busy us over the next half an hour or so. We know from Fisherian selection theory that in dioecious populations with separate sexes, where we just have males and females, in any organism, plants or animals, we expect, under most circumstances, a 1-to-1 sex ratio. Now, this can change in circumstances where there is strong inbreeding or strong population structure. I won't get into those complications in this talk, but I think for the most part, we can just go back to this basic idea that we expect in outcrossing populations a 1-to-1 sex ratio if there are separate sexes. In hermaphroditic populations, this basic idea translates into a 1-to-1 expectation for the sex allocation. So what does that mean? It means that hermaphrodites are allocating their resources to male and female function, and they should do so in such a way that half of their effort is allocated to their male function, half to their female function, roughly. This raises interesting questions because from the point of view of these transitions between separate sexes and combined sexes, we have an interesting shift in terms of sex allocation, from the point where individuals are in dioecious populations are deciding their sex allocation in terms of the numbers of sons and daughters they produce. In plants, that would be the number of male seeds and female seeds that then go on to develop into male and female individuals. And when that transition takes place to hermaphroditism, the sex allocation decision changes. It's no longer a question of the sex of the individuals that are produced as seeds by a mother, but it's the case of how much pollen is produced, how many ovules are produced, what is the reproductive effort that goes into attracting mates through the production of petals, and to what extent is that divided up between male and female function, and so on. So the sex allocation questions become, to a large extent, more complicated in hermaphroditic populations. It also raises the question as to what is the relative cost of the male and female function. We should expect, as I say, in an outcrossing population that the allocation to both male and female function should be equal, but there is has been a lot of discussion in the literature about the possibility that female function is a more expensive function for plants than their male function. How does that square up then with this expectation that in fact the two sexual functions should be invested in equally? I'll come back to that in a moment. And the other possibility that is raised by having a hermaphroditic strategy where plants have both male and female function is that sex allocation might more easily become condition dependent. In other words, a hermaphrodite individual can actually vary its resources allocated to male and female function depending on how examples access to resources, the types of resources that it has. So I'd like to explore these ideas looking at 3 different study systems that we've been using in the lab recently. The first concerns condition-dependent sex allocation in an alpine perennial herb, Anemone alpina, which is very common in the Alps in Europe. It's a perennial annual alpine herb. It's pollinated by flies. And the reason we were attracted to having a closer look at its reproduction is that it displays very large variation in its sex allocation. Some individuals only produce male flowers. Others produce female and male parts within their flowers, so they have hermaphroditic flowers. So we have a mix of potentially male individuals in the population, hermaphroditic individuals in the population, and what's particularly interesting is that the hermaphrodites also are incredibly variable in their sex allocation. And so on the slide, you can see on the right-hand side pictures of 2 flowers, and close up, one at the bottom is the male flower, which only has stamens, whereas the flower in the top right is a hermaphroditic flower, which has stamens that surround the pistils, the carpels in the center of the flower. And the numbers of stamens and carpels vary enormously from one flower to another within an individual and also between individuals. So this seemed to be quite an interesting model to address questions concerning the relative costs of male and female function and the condition dependence of decisions in terms of how plants allocate their resources. So I won't go into a huge amount of detail into what we've been doing here. This is work that's been conducted by a new PhD student in my lab, Kai-Hu Chen from Taiwan, who has been interested in looking at sex allocation questions in anemone. And I'll just present some recent data that we have on the basis of an experiment that Kai did, a very simple experiment where he simply manipulated the resource status of plants in 2 ways in a bifactorial experiment. On the one hand, he removed all the basal leaves of the plant. So the plant produces a type of rosette that obviously produces the photosynthase that support the perennial life history and flowering in this plant. And so there were 2 treatments here, those in which the plants were left unmanipulated and others in which all the leaves were removed. And Kai then had a look at how that affected reproduction the following year. And the second treatment was to remove the buds, in other words, these are the flower buds, before the inflorescence had been able to be produced. So it's effectively removing from the plant the necessity or the ability to allocate to reproduction. And the idea there is that this would increase the resource status of plants the following year. And what we find is perhaps not surprising. First is that for those plants, well, I should perhaps just say right up front that the bud removal treatment actually had no effect over the 1 year of this experiment. So I'll be presenting all the results from Kai's work here, but the 2 right-hand points here in the graph both correspond to the leaf removal treatment, where the left-hand points correspond to the controls. And we can see that where leaves were removed, the proportion of individuals the following year that did not flower, in other words, that presumably had insufficient resources to flower, increased. So we can see that leaf removal had an effect on the resource status of the plant such that it reduced the flowering rate. So what about the actual sex allocation of those individuals that did flower? And here I'm just presenting 2 results concerning on the left-hand graph, the pistil number, that's the female function, on the right-hand graph, the male function. And we can see that for those plants that flowered, there was actually no effect of leaf removal on the number of stamens in the flowers. It had very little effect, if any, on the male function of these individuals. But it had a strong, the leaf removal had a strong effect on the average pistil number. Those individuals that did flower flowered less as females than they did as males. And I think the implication here is that in some way, female function would seem to have been more expensive for plants. Plants with low resource status are choosing to invest less in their female function and more in their, well, not more in their male function, but less in their female function, and therefore their relative allocation to reproduction, their sex allocation becomes male biased. And that is something that we'd noticed from just surveying the populations, that the small individuals were those in which we found only male flowers. The large individuals with large inflorescence displays tended to be hermaphroditic. That might not be that surprising if one thinks that, well, of course, the female function is expensive. Plants have to produce seeds, they have to produce fruits. The investment takes place over a longer period of time. Of course, the female function is going to be more expensive. However, that sort of thinking doesn't really square up with our original expectation that plants should actually be allocating resources equally to their male and female function due to Fisherian sex ratio selection. And so that raises quite an interesting question. How do we actually understand this deviation in terms of what appeared to be differences in the costs of allocation, given our expectation of Fisherian selection? And I think the answer to this has to do with how one translates investment into these two different functions and the fitness that one gets under different conditions. So why should female function be more expensive? I think the answer to that is that it should not necessarily be more expensive. And indeed, we've done experiments which were similar to Kai's experiments on wind-pollinated herbs. And there we find the opposite, that male function is more expensive in the wind-pollinated herbs. What we found in those experiments, I won't show you the results here, but if you prevent males and females from flowering, the males become much bigger than the females. In other words, they are really able to invest much more in growth if they don't have to flower than females, suggesting a much higher cost to male function in wind-pollinated herbs. So I don't think we can conclude by any means that female function is always the more expensive function. And in any case, I think this idea of what is more expensive needs to be seen in the context of the fitness implications. And I'll just refer to some basic theory that it's quite old now, that was done by people like Charnoff, and this is some work by Klinkhammer, de Jong and colleagues back in the 1990s, investigating the potential or the implications of size-dependent sex allocation, where large individuals have more resources than small individuals. And what they showed on this graph is that, well, we see these two curves for male function and female function. It's the relative allocation to male function plotted on the x-axis, and the fitness returns from that allocation on the y-axis. And we see that for the male function, there's a very steep increase in fitness when plants have initially allocate small amounts to male function, and that then flattens off as the plants become larger or have more resources to allocate to their male function. We have a similar curve for the female function, but in this particular graph, the initial deceleration is much less than it is for male function. And what this does is it tends to favor individuals accentuating their male function when they are small, and therefore their female function when they are larger. And so I think this helps to understand why we get these differences in sex allocation as a function of the condition of plants. The next thing I wanted to consider in a little bit more detail now is the implications of male mating success specifically. And this introduces the idea of what we call Bateman gradients, which is the relationship between mating success or the effect of mating success on a plant's fitness. And this is an idea that's been thought about a lot in the context of animal reproduction, and surprisingly much less so for plants. The basic idea here is that if we plot mating success as a function of attractiveness, this might be the size of an inflorescence, or for female function, we might expect that a plant or a particular flower achieves its fitness quite quickly after it's visited by one or two pollinators, but subsequent visits by pollinators do not actually increase its fitness at all, because basically its ovules have already been fertilized. Whereas for male function, the fitness can continue to increase with every subsequent visit by pollinators, because individuals visiting the plant, pollinators visiting the plant, will be taking pollen off to other individuals elsewhere in the population. And so every visit has the potential to increase the plant's fitness from its male point of view. And so we might expect there to be an increased fitness with increased attractiveness in males, much more so than in females. This is a review that was done by Janneke et al. Some time ago, just to illustrate the tendency of many animal species to show positive Bateman gradients. In other words, situations where there is increased fitness to increased mating success for investment by males than by females. But as I say, this theory, Bateman gradients, have hardly been addressed at all in plants. And a former postdoc of mine who's now moved to the University of Montpellier, Jean Tonnerville, collaborating with me and Patrice David and Etienne Klein in France, had a look at how the reproductive success of plants might depend on their mating success in a wind-pollinated herb plant called Mercurialis annua, which you'll see a little bit more of in a moment. And there were two things I just wanted to say about this. The first is that we use microsatellites to actually to see how the numbers of mates actually led to reproductive success in Mercurialis annua. And Jan found that for female function, there was no real relationship between mating success and reproductive success through female function, as one would expect under the Bateman's law. But there was a very strong relationship between mating success and reproductive success in males, just as we would expect. What I will not show you in any further detail, but I should mention, is that the results were actually quite complicated, because Jean looked at how the architecture of plants affected their male versus female function. And we find that many of the secondary sexual traits that differ between males and females, such as the branching architecture or these interesting inflorescences that you can see on the right-hand photograph there in males, the length of that stalk had a large effect on male mating success, for example. So there are some interesting complications in terms of how secondary sexual characters that go beyond just investment affected the reproductive success of individuals. What Jeanne was effectively showing in her experiment was a component of sexual selection. So if I back up just for a second and we look at these two photographs of the male and female plants, the male on the right, the female on the left, we think that these differences between males and females are the result of different Bateman gradients between male and female, between males and females, and that there is very intense competition amongst males to sire progeny. And so many of the traits we see in males are probably the result of intra-sexual selection, competition between males to fertilize a limited pool of ovules. And I think that we could probably generalize this to say that many of the traits that we see in plants and the amazing floral adaptations that many insect-pollinated plants have are very closely related to selection or the basis of sexual selection. So, for example, we've seen the example of the wind-pollinated plant Mercurialis annua down on the bottom left here, where males and females have really quite different morphologies as a result of sexual selection operating probably mainly on males. The top left, there's a little scrophularia in which the anthers are displayed or are exposed one by one. These flowers each have 4 anthers, but they don't present their anthers all at the same time. Each anther comes out on successive days, and so the flower is actually open for a number of days, and each day the flower is dispersing a little bit of its pollen. That sort of adaptation, which we call a pollen dispensing mechanism, is very common in plants and enhances male function, whereas the female function may actually only be receptive for a single day. So for the rest of my talk, I'd like to just quickly run through a few implications of separate and combined sexes. First concerns norms of reaction to mating opportunities. Second is the evolution in response to changed mating conditions. So this is genetic evolution. And finally, if I have time at the end, I will show a few results from some work that we've done on gene expression in plants that have evolved separate sexes and looking for differences in sexual in gene expression between males and females. So the first study was conducted by a student of mine, Guillaume Cossard, and he was interested in looking at the potential for leaky sex expression in males and or females of this plant Mercurialis annua. The idea here is that males and females may occasionally produce flowers of the opposite sex. And of course, that sort of ability must be the basis of any transition between combined and separate sexes. So these transitions between hermaphroditism and dioecy. This is a plant with an XY system of sex determination. And we know from the field that males and females occasionally do produce flowers of the opposite sex. So what Guillaume wanted to know is how frequently that occurs and to what extent it's actually a function of the mating environment of the plants. And so this is what we actually find in the field. This is the distribution of flowers of the minority sex. So in the left, we have females that are producing a few male flowers. Most females are not producing any male flowers. Of course, they're females, but some of them do produce male flowers. And similarly, the males occasionally produce female flowers and fruits. And Guillaume did an experiment where he basically he set up 6 populations. 3 of them had 50% males and 50% females, as we find in normal populations in the wild. And in 3 other populations, he simply removed all the males. And so the females would be very strongly pollen limited. And the question was, to what extent do those females increase their allocation to male function, to male flowers, as a plastic response to the absence of mates? And the results are very clear. we're looking at the proportion here of females with male flowers on the x-axis in the first graph, and you can see that in those populations in which males had been removed, plants actually produced more male flowers. They were sensitive to their mating environment and responded accordingly. And the mean reproductive effort to male function was also much greater in those females where the males had been removed. So we see there is a plastic effect here on the sex allocation in a dioecious plant tending towards a type of hermaphroditic strategy in the absence of mates. The next thing, so this is just a summary suggesting that plants may be sensitive to whether their ovules are being fertilized. Is that the mechanism? We don't actually know how plants are detecting whether they have pollen available, but we think that it's possibly something that will be detected at the point of fertilization. Guillaume then went on to use the same experimental setup and to see to what extent the variation in male flower production by females could actually be material for a genetic response to selection over a number of generations. So the question is basically, can this variation in male flower production by females actually lead to the evolution of full hermaphroditism via natural selection? And so again, we've got the same experiment with the 3 populations with males and 3 populations without males. And the prediction was that selection for increased male flower production would occur in the selection lines. Why? Because firstly, the females are pollen limited. And so any females that produced male flowers would be able to produce seeds by self-fertilization. And secondly, and I think much more importantly, because these were dense populations, plants that produced male flowers would not only be able to self-fertilize, but they would be able to fertilize other individuals in the population. In other words, there would be strong competition amongst females for mating success as males. And these are the results. We did this experiment over a number of years, and we measured each year the proportion of females with male flowers and the fold difference in male flower production. And on the left-hand side, the 2 graphs on the left-hand side show what we were seeing in the field. So each year we measured what was going on in the field. And so what we see there is a mix of a plastic and potentially genetic response. And we see that those plants without males evolved to produce more male flowers, and there were more of them doing that. And after 4 generations, we took all the progeny and grew them all up in a single common garden so that we could exclude the possibility that the difference was simply due to phenotypic plasticity. And we see here that there is a very strong response to selection that is not plastic in the common garden. Plants that were growing and evolving in the absence of males evolved a very substantial male function. And these are pictures of the sort of phenotypes we see. On the left-hand side is a normal female, and on the right-hand side is the phenotype that we were seeing at the end of 4 generations of selection. And it's covered in male flowers, which are indicated by the arrows. Interesting, at generation 6, so we've continued this experiment over a number of further generations, and at generation 6, we see the invasion of a female sterility mutation in one replicate. And this mutation basically is associated with massive increase in male flower production of those individuals. So we're effectively seeing the reinvasion of a type of male into these populations after the removal of males at the beginning. And this shows these males, the female sterile phenotype with relative male allocation at 1, and it's way more male than any of the other individuals in these populations. We are now seeing this female sterility mutation in all 3 of the replicates without females. So it's probably was a mutation that was at a low frequency initially in the base population, and the experiment is continuing as we see this experiment, as we see this phenotype increasing in frequency in all three of the replicates. So to summarize, we find that females of Mercurialis annua rapidly evolved a male function when males were removed. And we think that this is the result of selection, both for overcoming mate limitation, but much more importantly, because of the huge siring opportunities that individuals had when males were removed from these populations. The main difference between males and females or hermaphrodites, the evolved hermaphrodites, is the possession of this interesting inflorescence stalk that one sees in the males. The peduncle is a Y-linked trait. The possession of a Y chromosome is directly linked to having that particular characteristic, that inflorescence structure. And we find that pollen dispersed from those inflorescences on males has a 60% siring advantage over pollen produced by females that do not produce these long inflorescence stalks. And so we're interested in the evolution of this particular trait. This goes back some time in the lab. We looked at the variation in sex allocation in the genus, and in particular annual clade of the genus. And what we can infer is that the basal lineages of Mercurialis annua, or the Mercurialis annua complex, were dioecious. There was then a transition to dioecious and perennial, and there was then a transition to an annual life history that then diverged into a number of different species. I'm showing two of them here, Mercurialis hueti and annua. Both of them still have 16 chromosomes and are dioecious and diploid. The next thing that we see is that the annual species, Mercurialis annua, went through autotetraploidization, doubling of the chromosomes, and this corresponded to a shift from dioecy to monoecy. So something very similar to what we saw in our selection experiment. Then, interestingly, there was a hybridization event between Mercurialis hueti and the polyploid populations of Mercurialis annua, leading to an interesting sexual system called androdioecy, where we have males co-occurring with hermaphrodites that are females producing male flowers as well. These populations are hexaploid. And we assumed that, or predicted that, the male inflorescence and the Y chromosome in these hexaploid populations must have come from Mercurialis hueti, because the female parent in that cross was Mercurialis annua tetraploids, which do not have males and do not have Y chromosomes. So a student of mine, Jörn Gerchen, looked into this with a postdoc, Paris Veltzos, and they basically took Y chromosome loci from a number of different species and looked at the phylogeny of the Y chromosomes. And what they found was quite surprising. The Y chromosome in the hexaploid annua did not come from hueti, from the sister species, but actually had been derived and introgressed from an outcrossed species, from a basal species that was perennial. So it's much more distantly related. And so we see evidence here of the introgression of a Y chromosome from a much more distantly related species into populations that then could evolve this weird sexual system, androdioecy. So the Y chromosome seems to be moving around between different species here. If we look at the distribution of these sexual systems in the Iberian Peninsula in Spain, we find that these are the hexaploid populations. The androdioecious populations are found all around the Iberian Peninsula, where you've got males and females found in these populations. But we find that there is, in this region in southeastern Spain, a new type that we hadn't noticed originally, where the hermaphrodites have in fact evolved the inflorescence stalk that confers increased reproductive output or increased reproductive success. And so we have basically two scenarios here. We have a large region of the Iberian Peninsula which has males that are producing male flowers without the floral or the inflorescence stalk. And then in a region in eastern Spain, we find that the females have actually gone on to evolve an inflorescence stalk which confers increased siring success. And so this is what we think is going on in the evolution of sexual systems in this species complex. The original state is separate sexes, with males and females on separate male and female flowers on separate plants. Then there was the evolution of monoecy, probably as a result of selection for reproductive assurance during range expansion or through colonization in metapopulations. Once these populations have established, there is very strong competition amongst individuals for increased siring success. So there's strong selection to increase allocation to male function, but also to improve the dispersal of pollen. And we see that the populations have responded in two different ways to selection for improved outcross siring success. Either we have the evolution of male inflorescences on these hermaphrodites that improve the siring success of the pollen that's dispersed, or we have the reinvasion of males into those populations. So we have a dimorphism that re-evolves probably through migration of males from the base population. And the existence of males in these populations seems to suppress the evolution of improved male siring success in the hermaphrodites, which effectively are maintaining mainly a female function with relatively little allocation to their male function. So we see here in two different parts of the species range, two quite different outcomes of selection for improved siring success, either a polymorphism where males co-occur with modified females, or a male-like form evolves within the context of hermaphrodites. So the pedunculate inflorescence promotes the siring success in Mercurialis annua. It's Y-linked. It probably exemplifies a sexually antagonistic trait that confers increased fitness on males, but probably either does not affect female fitness or it reduces female fitness, and that's something we're looking to at the moment. And it's moved, the Y chromosome seems to have moved among species via polyploid hybridization. Let's see how I'm going for time. I've got just enough time to show you some recent results that we have from studies of sexual dimorphism and gene expression in the genus Leucodendron in South Africa. So this is a study that I've been doing with Matthias Schaagman, a postdoc in my lab, and a collaborator in South Africa, Tony Rebello. And this is the genus that we are studying. It's a remarkable genus in many ways. Firstly, it's a large genus in the Cape Floristic region of South Africa that has about 80 species, all of which are dioecious, and that's quite unusual in plants to have a large dioecious clade. The other thing is that there seems to be an XY sex determination in all of the species. We've looked at a number of them now, and they all seem to have the same XY sex determination system. But what drew us to this system in the first place was the very wide variation in sexual dimorphism that one sees. And I've shown just an example of that here with one of the species, Leucodendron rubrum, which is wind pollinated. Some of them are pollinated by insects, others are pollinated by wind. And you see here very big differences in the morphology between males and females. And we were interested in understanding why sexual dimorphism has evolved in some species but not others. I won't have time to go into that question. But this particular study concerns patterns of gene expression. And the question is really quite a straightforward and perhaps rather a banal one. Do we see differences in gene expression that correspond to the massive differences in morphology? And specifically, do we find that species that have very strong sexual dimorphism, do they have stronger sex-biased expression of genes than those that are actually very similar in their morphology, where the males and females really have the same leaf size, same branching architecture, and so on? And the genus is provides wonderful material to look at this, because sexual dimorphism has evolved independently several times within the genus. This is the genus here, and you can see here just on this simple mapping of species that have no dimorphism, low dimorphism, or strong dimorphism, that basically strong dimorphism is evolving independently again and again. Recall that all of these species have separate sexes, but only some of them show strong dimorphism, and this is something that is convergent in different lineages across the clade. And so we asked whether convergence in sexual dimorphism is associated with convergent patterns of sex-biased gene expression, and how those patterns of gene expression have evolved. In animals, this question has been addressed by looking at a number of different, quite distantly related birds. And this is work that was done in Judith Mank's lab. And we see that the proportion of male-biased genes were related to sexual ornamentation. So those species that had strong sexual dimorphism and ornamentation also had strong male-biased gene expression. We were looking to see if we found the same thing within a clade of angiosperms. So the study was based on 5 different comparisons between strong dimorphism and low dimorphism, independently sampled across the phylogeny. So we've sampled patterns of gene expression across 10 different species and an outgroup, a hermaphroditic outgroup, and the sampling was structured in pairs: a strongly dimorphic species versus a weakly dimorphic species, or species without any sexual dimorphism that we could see. And we asked, do those species with strong sexual dimorphism in terms of their morphology also have more sex-biased gene expression? And we were surprised that actually there's absolutely no correspondence between these two different measures of sexual dimorphism, morphology versus gene expression. I won't go into any of the details here, but I think the graph makes this very clear. These are the number of sex-biased genes plotted on the right-hand side of the figure against the phylogeny in the different species here. And you can see that, for example, Leucodendron dubium, the one in the center here, has the largest number of sex-biased genes, even though it's probably the species with the least sexual dimorphism for leaves. Whereas Leucodendron rubrum, its sister species within our phylogeny here, has the greatest level of leaf dimorphism in the genus, and yet it has a much lower level of sex-biased gene expression. The other thing we found is that although we find many genes that are sex-biased, only 163 genes across the genome are sex-biased in more than one species. And of these, 61 genes are male-biased in one species and female-biased in the other. In other words, there are even genes in which in some species the bias goes towards male function or towards male bias, and in others it goes towards female bias. So we're getting a picture here of most of the sex-biased gene expression being very recent in expression. It's not something that has evolved early in the diversification of the genus and has been retained. The sex-biased gene expression we find in extant species has evolved along the lineage since divergence from the sister species in our sample. So sex-biased gene expression is evolving and turning over very rapidly. And so the final question we asked here, and this is the last point I want to make, is what are the rates of expression evolution in sex-biased genes? We can see that there are genes that have become sex-biased in our sample and those that show no evidence of sex-biased expression. Do these two different classes of genes evolve differently in terms of their expression levels across the genus? The base of gene expression for all pairs of species and the levels of divergence in terms of their non-synonymous sequence divergence as a way to calibrate the extent to which they were evolutionarily diverged. And the result was quite clear that sex-biased genes are evolving their expression levels much more and significantly faster than non-sex-biased genes. So genes that are sex-biased are changing their expression levels more than those that are not. And we then asked, well, is that because genes that are sex-biased are changing their expression levels as a result of sexual selection, which is one of the thoughts that people have had to explain these patterns in animals. For example, the study on birds emphasized this possibility that if there, in those cases where there is strong sexual dimorphism, there's also strong sexual selection, and that might have an effect on accelerating rates of expression evolution in the genes. But in fact, that's not the case at all in leucodendron. And the reason is that the genes that are sex-biased were actually already evolving more quickly before they evolved to become sex-biased. In other words, the increased rates of evolution in gene expression preceded the evolution of sex bias. And so what that seems to imply is that sex-biased genes are recruited from genes that are less constrained in their expression levels and may have much less to do with selection or sexual selection than one might have thought. In other words, there may simply be a lot of neutral divergence in gene expression between males and females simply because these are genes that can actually change their expression levels rapidly without major fitness implications. So that's all I have time to present. I just end by acknowledging the funding sources and the various different people I've mentioned along the way who contributed to this work. Thank you very much for inviting me to speak again. I'm happy to take any questions.
I have a trivial question. I was wondering how mean reproductive effort towards male function was calculated. In some of the earlier graphs, you had shown that for the female plants.
For plants, so we calculated. Was this in the female plants of Mercurialis annua, where we were looking at the?
Yeah.
Yes.
Yeah.
We calculated it in two ways. One was just in terms of counts of male flowers, and secondly, as the biomass allocated to male flower production. So we removed all the flowers from the plants and measured their biomass. Now, of course, a measurement of biomass to male allocation or male flower production is not at all equivalent to or comparable with allocation to female function. what we find is useful in our system is that we can use the allocation of resources by males to male function in a way to calibrate the importance of male function in females. So, for example, instead of just simply measuring male function in absolute terms in females, we can relate that to what a pure male would be doing. So we can measure the male function in females on a scale from 0 to 1, where 1 is what a male would be doing, and 0 would be what a pure female would be doing. And so we have a relative measure there as well.
So Prasad has a question. How does one measure? This is relatively easy to do in lab systems of animals, but how does one do this in plants?
So you raise a very interesting and important question there, Prasad, which we sort of had long discussions about how one actually does this, because what we were ultimately forced to do was to actually use microsatellites to measure mating success. And so we have an array of individuals in which we have all individuals genotyped, and we then assign to each seed that we sample a particular father. And so for a particular female individual, how many mates does that individual mate with? Well, we obviously have an estimate of the number of fathers that are assigned to its seeds from a small sample of the seeds that individual produced. So our estimate then is clearly a very conservative estimate, that a plant that we might have sampled 30 seeds on a particular plant and assigned a certain number of mates to it. But of course, the plant can produce many more than 30 seeds and may well have had more mates than we actually measured. And so there is a real constraint there in terms of what one actually can do in a plant population. So our mating success it's a conservative estimate of how many individuals that particular plant had mated with. Similarly, for the male function, one can see how many separate females a particular male mated with by taking a sample of seeds from all the females in the population and assigning paternity. So in a sense, what we're doing is our estimate of mating success is actually quite close to, or is closer to an estimate of the male component of fitness than you would really like to have, and that you would be able to use in an animal population where you can actually observe matings occurring. Because we're of course not including any of those mating events where a pollen grain actually did settle on a stigma of a female. You would normally say that was a mating event in the context of a type, in an animal system, for example, and that must also be a successful mating event in a plant population. But we are missing those entirely from our study. We don't know. We're only looking at those mating events that actually led to successful fertilization. So that is certainly a constraint in what one can do in this sort of system.
I also was struck by how fast you had a response in those experiments. Is that surprising, or are these responses generally fast in these kinds of systems?
It was very surprising when we first saw it, but of course, when you start thinking about what must have been going on in these populations, perhaps it's not so surprising. If you imagine a population of females in which all the males have been removed, our populations had about 200 individuals in them. Any individual that produced any male flowers in that population would be able to produce its own seeds by selfing, but much more importantly, it has access to all the ovules produced by all the other individuals. So the fitness of a male flower-producing female is absolutely enormous. And when we model this, we can see that actually the strength of selection is very strongly dependent on how many males there are in a population. As soon as you have males in a population, the strength of selection on females is reduced enormously. But you take the males away, there's extremely strong selection on females to increase their male flower production, even to the extent that plants that sustain a female sterility mutation and lose their female function, if that translates towards increased male flower production, that mutation will spread very rapidly. And I mentioned that at the end of that section of my talk, that we're seeing this female sterility mutation spreading very rapidly in all three of the replicates, presumably because it confers increased male siring success. So that's probably been the biggest surprise from this experiment, that not only are we seeing the evolution of basic hermaphroditism, but the hermaphrodites have not been able to respond nearly quickly enough to a female sterility mutation. And it's going to be very interesting to see how these populations evolve now. Will the invasion of these female sterile individuals, will that, in a sense, put a stop to the selection on the females that have maintained their female function? So we're sort of getting a divergence now in sex allocation between two different phenotypes. So that'll be interesting to see over the next few years.
John, what is the pattern of distribution of these varieties of plants? Andreaceae, monoecious, hermaphrodite, all these are they sympatric, or they're scattered in different places?
Within the Mercurialis lineage we were talking about?
Yeah.
That's a very good question. There are, in general, they're allopatric. So the dioecious populations occur throughout Eastern, Central, and Western Europe up to about the Pyrenees. They go across the Pyrenees into northern Spain. The monoecious populations are found then around the Iberian Peninsula, and there are two contact zones between the dioecious and the monoecious populations in northern Spain, but there's no overlap. And in fact, at those two contact zones, there's a very interesting interaction occurring where the populations are mating with, well, there's mating within occasional sympatric populations, but the progeny produced between these are sterile, and you get very strong pollen swamping in one direction, leading to a shift in the distribution of the systems there. But in general, we see these systems maintained in different parts of the European range. The two different strategies that we found in the hexaploids, they do overlap in their distribution. We have some populations in which we have males and females with a male function, but without the inflorescence, the male inflorescence morphology, and then other populations which lack males and have the male inflorescence morphology. And they tend to be found within the same regions. We've not found any in the same actual populations, but they tend to be found in the same regions. So there is a sort of a regional sympatry there, but they don't tend to occur in the same populations.
John?
Yes.
Well, one last question. When the transition is taking place from monoecious to dioecious, and as you said, there are transition phases. So
yes.
During these transition phases, do you get hybrid zones?
I'm hesitating because there are hybrid zones now between these between the dioecious and the monoecious populations. But I think, well, in fact, in that particular case, the hybrid zone is a secondary hybrid zone. We know from patterns of genetic variation across Europe that the populations in the hybrid zone have both diploid populations or the dioecious populations have come from Eastern Europe, and the monoecious populations have come from Northern Africa, and they've met at what is now a secondary contact zone. They are hybridizing, but as I said, the F1 generation plants are completely sterile, and we don't see any evidence so far for any gene flow across that boundary. I don't think we see any primary hybrid zones in the sense that you might be suggesting.
What is the mechanism? I think this is from Prasad. Plastic development of male flowers in M. annua are the females of pollen, and if so, does one see fewer male flowers earlier in the season and more male flowers later in the season?
If I heard the question correctly, the first was, what is the mechanism of male flower production?
Yeah, are the females sensing the absence of pollen?
That's a very big. That's a pressing question that we have. How do the females decide to produce male flowers? As we see, there is a plastic effect, and it seems that the presence of male flowers in the population, so the presence of males in the population, tends to reduce the tendency of females to produce male flowers. We certainly find that in the ancestral dioecious populations, and we think that it's most likely to be sensed at the level of seed sets. Plants that are not setting seed and producing fruits because their flowers have not been pollinated, that would be a very clear signal to the plant that there is no pollen in the population, and that an ability to produce pollen in those circumstances would certainly be strongly favored. We don't know now, of course, in the populations that have evolved a large male function, how much plasticity there is, because it's extremely difficult to control the mating environment once the plants are producing pollen. So I suspect that there is probably still a component of plasticity there, but it's difficult to pin down because we can't manipulate the mating environment so carefully there. I might also point out that just from the point of view of the developmental genetics of male flower production in these populations, the females that are producing male flowers are XX individuals. There's no Y chromosome. And so if you regard pollen production and male flower production as a primary sexual characteristic, then it's perhaps surprising that it's not associated with the Y chromosome. You might expect that the primary sexual trait, male flower production or pollen production, would be something associated with the Y chromosome, but it's not. It's somewhere else on the genome, and we haven't tracked it down yet, but it's not Y-linked. However, what is Y-linked is the production of the stalk-like inflorescence. So that is a secondary sexual trait, and it's the secondary sexual trait that is Y-linked, but not the primary sexual trait. Clearly, there will be somewhere on the Y chromosome a switch between male and female function, but the actual genes required for producing male flowers must be scattered all over the genome. I suspect they could even be on the X chromosome.
Okay. Thank you so much, John. This has been such an interesting talk. Thanks again.